Welcome to Cancer Lesson 3.4. How does DNA damage allow cancer cells to cheat death? The goals of this lesson are to explore the concept of cell aging and how aging affects DNA, and apply the concepts of telomeres and telomerase to cancer cells. At the end of the lesson, the students should be able to explain what a telomere is and how it protects chromosomal DNA during mitosis, explain how telomere shortening limits how many times a cell can replicate, and explain the function of telomerase, when it is normally expressed, and how its expression changes in cancer. We will achieve these goals by engaging in an interrupted case study exploring how inhibiting telomerase could cause cancer cells to die. To prepare for this lesson, you'll need to review the following key scientific concepts that will be presented throughout the lesson. Each time a chromosome replicates, it becomes shorter, but the genes are not damaged because the chromosome is capped by a protective stretch of repetitive DNA called a telomere. When telomeres become too short to protect the genes, the cell goes into crisis and dies. The enzyme telomerase prevents telomeres from shortening and is expressed in cancer cells, so cancer cells do not die. You can review this scientific content in the background reading provided for you in the teacher primer, the teacher manual, and the student workbook. The teacher primer provides in-depth knowledge about the scientific content presented in this lesson. The teacher manual or lesson plan provides a minute-by-minute explanation of the lesson structure. The student workbook provides additional explanation for students. Be sure to print the lesson worksheet and the homework worksheet. The key points of the do now are that telomere length controls how many times a cell can divide. When the telomere is gone, the coding region of the chromosome will be damaged, so the cell enters crisis and dies. Cancer cells activate the expression of the telomerase enzyme, which preserves telomere length and prevents crisis. We begin the do now by considering what aging means at the cellular level. As we age, cells must replicate. After a finite number of divisions, cells die. To replace these dead cells, stem cells divide and differentiate, but even stem cells do not divide indefinitely. As we age, the reserve of daughter stem cells becomes smaller. Each time a cell divides and replicates its chromosomes, the end of the chromosomes gets a little bit shorter. The video nicely illustrates why this happens. Luckily, the chromosome doesn't lose its genes. Instead, the protective ends of the chromosomes, called telomeres, get steadily shorter. But at some point, the telomeres become so short that the next time the cell replicates, actual genes will be damaged. At this point, the tumor suppressor gene p53 is activated and tells the cells to stop proliferating. This is called senescence. But what if the p53 gene is mutated and inactivated? It cannot tell the cell to go into senescence, and instead it goes into crisis. During crisis, the chromosomes fuse together and the cell eventually dies through apoptosis. So crisis is actually beneficial because it enables the body to limit the replication of cells that may be severely abnormal because p53 is inactive. But some cells are able to avoid crisis by using an enzyme called telomerase, which replaces the ends of the telomeres at every replication cycle. So telomerase can prevent a cell from aging normally. In fact, it becomes immortal, like cancer cells. In fact, 90% of cancer cells express high levels of telomerase. They are able to do so because the promoter region of the telomerase gene has mutated, so the gene is always switched on. Because of telomerase, they can replicate even though they are abnormal. The key points of the activity are that if telomerase is inhibited in cancer cells, telomeres will shorten each time the cell divides. This means that telomeres cannot be replaced. The cancer cells should be able to undergo crisis and die normally. We'll get there by having students complete an interrupted case study on inhibiting telomerase. In the lesson activity, students will consider an experiment that looks at the results of inhibiting telomerase in cells and in mice. Students should critically think about the results of the experiment, which may be surprising. Counterintuitively, mice treated with the telomerase inhibitors actually made tumors. 90% of the cells in these tumors did express telomerase and were killed. However, 10% did not and could still divide without dying because they were using another pathway that didn't involve telomerase. But why were these tumors larger than in mice that were not treated? The telomerase inhibitor selected for cells that didn't need telomerase because they used an alternative pathway to grow. In this case, those cells happened to have a more rapid growth rate than the original telomerase-expressing cells, and hence formed a large tumor. The 10% of cells that could continue to proliferate and become immortal without expressing telomerase used a second mechanism called the ALT pathway for alternative lengthening of telomeres. The final two slides recap the role of telomeres in normal cell aging and the role of telomerase in allowing cells to avoid crisis and become immortal. For homework, have students read an article about a woman who lived to be 115 years old. One of the most common questions students ask is, what is the difference between senescence and crisis? Senescence is what normally happens when telomeres become so short that a cell can't divide safely. It just stops dividing. Senescence is normally activated by p53. Crisis occurs when p53 is mutated. Now the cell tries to divide once more, and in the process, the chromosomes fuse together. At the end of the lesson, collect the lesson worksheets to assess students' explanations of the function of telomerase when it is normally expressed and how its expression changes in cancer. This lesson introduces how telomeres play an important role in the normal process of cell aging. We will build upon this concept in future units to understand how cells can evade normal processes and become cancer cells. Don't forget, if you have any questions, concerns, or feedback, to let us know. You can contact any of the CTSE team members and we'll be happy to help you.